Hello lovelies, in this video we're going to look at the development and understanding of genetics. So how we know what we know now and how this is still a really developing field.
Development of the understanding of genetics. We owe a lot of our understanding of genetics to a monk called Gregor Mendel, who studied the inheritance of characteristics in pea plants. He was doing this long before the discovery of DNA or genes. He didn't know what genes were at the time, but he was aware of these things that he called units. He realised these units were passed on to offspring from parents and that they caused characteristics that were distinct to be inherited. At the time, it was thought that they were blended when parents produced offspring. So here's a homologous pair of chromosomes and some genes or units or characteristics that Mendel would look at. So whether the shape of the peas was wrinkled or smooth, and whether the colour of the peas was green or yellow. He observed that pea colour and pea shape are different characteristics that are inherited separately and have no effect on each other. So offspring could have a mixture of these different types of characteristics. They could have wrinkled yellow or smooth yellow peas, or they could have wrinkled green peas or smooth green peas. So it didn't really matter. There was a mixture there of inherited characteristics, and they could tell that these units were inherited, but also that they were passed on distinctly. So either the colour of the pea or the characteristic of the pea shape, they were separate units to Mendel. So if they were blended, you'd get like semi-smooth, semi-wrinkled, half green, half yellow pea. And that wasn't the case. And he was able to observe that. He also was able to recognise that characteristics could disappear and then reappear in later generations. We now know that these are recessive alleles for genes. So this is the one we looked at when we looked at inheritance. So we had our white flowers and our red or pink flowers. And in the first generation, when we mixed the white and the red parents together, we got all red flowered offspring. So the white phenotype was missing in this generation. But then when we took those offspring, so the heterozygous red flowers, and we bred them again, and Mendel did this so that he saw it happen, he would get a second generation and suddenly the white phenotype came back. So we have kind of the original parents, and then the first generation is all red, and then in the third or second generation, we have that white phenotype popping back up again. And we know that obviously this is caused by a recessive allele now, and so that's why those parents were carriers of the recessive allele for the white flowers, and so that's how it kind of disappeared and then popped back up again. So this is what we now know and how we now understand recessive alleles. At the time, they didn't have the name for that, but he was able to say that he saw these observations and saw them consistently over breeding lots and lots and lots of different pea plants. He published this work, so his comments on this idea of units, this idea of inherited characteristics from parents to offspring, and this idea of missing characteristics popping up later on in different generations. He published it all in about 1865, so the mid-19th century. Mendel's work wasn't recognised or accepted by other scientists at the time, around the mid-19th century, because of many reasons. They didn't really understand what he was trying to say, because without the knowledge of genes, chromosomes, and all the things that we know now, it wouldn't have really made much sense to try and figure out how to explain those observations that he was seeing. Not many people read the small journal that he published it in, so it was quite an obscure publication, quite small, not widespread, and not read by many people at the time. Again, he could not really explain the science behind his observations. He couldn't explain what units were. He couldn't explain how the patterns that he saw were happening because DNA, genes, chromosomes were not known about at the time. And many people at the time still believed in this idea of blended characteristics, this idea of the sperm and the egg fusing or gametes fusing and creating this blended organism that had bits of both parents, but not distinct, separate characteristics from each one being inherited. The importance of Mendel's work wasn't realised until way after his death. This was when improved microscopes and staining techniques allowed chromosomes and their movement in mitosis to be viewed. This was around the late 1800s or the late 19th century. In the early 1900s, it was realised that the chromosomes behaved similar to Mendel's description of units. This led on to the idea of genes being located on chromosomes, and this idea that it was those genes on the chromosomes which led to the particular characteristics. In the mid-1900s, the structure of DNA was determined by two scientists, James Watson and Francis Crick, in Cambridge. They built on this work using other images taken by Rosalind Franklin and Maurice Wilkins to determine that the shape of DNA was actually that twisted double helix. This was then used to discover the function of genes and how they code for proteins, something we now call gene theory. And finally, the most recent developments in human genetics are that we sequenced the human genome in the 2000s, and this led to gene therapy and other potentials for things like personalized medicine, so having drugs that are tailored to your genome.
Ouch! This is why in some videos I explain scratches.